And so we do have these seminars coming up. You know, they're just volunteer. I'm just posting them because it's something that's happening, and it's very interesting stuff that's happening. Oh, let's do this one time. Thank you. And so here we go with this. And I have your exams back. I will hand it back at the end of class. You'll have a circled score. I made an answer key video, and I also made a grade distribution. And so I'll post those on Brightspace, and you'll be able to go to the grade distribution, take your circled score from your exam, and see what your letter grade was for this exam. Each student is represented by asterisk on that, so you'll be able to see overall anonymously how people did. And we'll move forward from there. You want to go through the answer key video. After the first answer key exam, we had an assignment. We won't have an assignment. This time I said we're going to be done with assignments except your term paper. And but I super encourage everybody, no matter how well you did, to just watch the whole thing. Because I try to have it be a learning experience. And even if you got full credit on some questions, there could be a way to answer it better a lot of times, And I'll give you an example of that right now. No, I won't, because I can't quite remember the full question. But I'll give that back at the end of class, and then you'll be able to see that, and we want to follow up with that, right? We're preparing for the final. The final is going to look like the first and second exams. You got the answer key for those exams. You want to make sure you're solid on those things, because we'll loop through that stuff for the exam, but also build up more. We're building up more all the time. So what should you do this week? So you want to read the last chapter of the book that we're going to read. We'll cover a lot more new material, but you just won't be required to look at the book. And it's about the nucleus, and then you're working on your term paper. So that's where we're at. And does anybody have any questions about anything at all, of any kind? How many weeks are left in the semester?
Maybe 2.
I don't know, who cares? But I know this, I forgot to put this up here. Our exam is like 2 weeks away, or something like that. From Tuesday, something like that. And it's May 6th. And it's in this room, and it starts at 8. It'll be the same style as our first 2 exams, so you won't need 2 hours to do it. If you happen to want to just get in here a little bit later, just come in quietly, go to the bathroom, turn off your phone, put it out, take the exam. And we'll talk about that more, but that's what we're getting ready for. It's just the same thing. And it's not like, oh, let's ask a general question because we're kind of wrapping things up. No, it's just like, what's this, what's this, what's this? Just the same format all the way back to the muscle differentiation videos. Yep. All right, so that's where we're at. And so we're in this section of the book that really is a cell biology section. The idea of we got to look at the cell and components of the cell to really understand the situation. And so in the nucleus, we got to look at the nucleus. And our number one thing to do on that is to look at that nuclear pore. So the nuclear membrane that surrounds the nucleus has a gated entrance in and out of the nucleus, and it's through nuclear pores only. Some small molecules can just freely diffuse through there, but big molecules like proteins, RNA, and DNA, they have to be let in and out of that. So we were learning about that on Tuesday, and we're just going to finish that up and keep rolling through chapter 10 like that, okay? And so, for sure on the exam, there'll be a thing of just lists, make a list of proteins that would have to have a nuclear localization signal. So we talked through that on Tuesday, and that's to get in. And who's going to help them get in? Well, important is going to help them get in. That important is what's going to bind to the nuclear localization signal, and it will then escort its cargo into the nucleus. Then it will release its cargo, and it will exit the nucleus through that exportin pathway. And there's a whole class of different kinds of importing and exporting protein, and we're just going to learn those 2 terms. The book has a list of them, and you just don't need to go there for some of the other ones. And so that explains this thing that we started our course with is, FOXO is a transcription factor. To do its function, it has to get into the nucleus. It can't get into the nucleus when it's phosphorylated because it's sequestered in the cytoplasm by 1433. And how does 1433 keep it out of the nucleus? It blocks the nuclear localization signal that's always part of FOXO. Proteins that have nuclear localization signals have it as part of their primary amino acid sequence. They just were made with it. But it could be covered up, and that happens with heat shock transcription factor as well. So we have a couple examples of that, of regulating the entry into the nucleus by regulating the nuclear localization signal availability for binding by proteins like important. As a result, we have this story with FOXO where, oh my gosh, the FOXO gene is expressed. It's a coding gene, it codes for FOXO protein. The FOXO protein, it's made, and but it doesn't get to do its job. So it's just as if the gene didn't get made at all. The regulation of the gene's function is all the way downstream at the level of the protein, and we call that post-translational regulation. The protein was actually synthesized, but it's still being regulated. And there's just tons of that. We learned tons of examples of that in our course of proteins being modified in one way or the other. That's all post-translational modification, and it's a big part of the story of how the genome plays out the information over time. Remember back at the start of the course, we picked muscle differentiation. We said, this is a pretty good story. We're just going to zoom in on these three genes. MyoD codes for a transcription factor that kind of immediately goes back into the nucleus and binds to the enhancer of myogenin and activates the expression of myogenin. And the myogenic protein gets made, and it kind of immediately goes back into the nucleus, and it binds to target genes and makes target genes express these muscle proteins, and we get muscle differentiation. And that was a really nice start, because you kind of have these dominoes falling. MyoD knocks down myogenin, knocks down muscle genes, and that's satisfying. And then we said, well, actually, though, in that loop that we were talking about on Tuesday, the idea that, well, these transcript factors are all proteins, so they must have a gene that codes for the protein. So MyoD must have a MyoD gene in the nucleus, and it's got to be in the nucleus. That's where the genes and the DNA are. So we get transcription of it, we get RNA processing from the primary RNA transcript to the mature RNA transcript that's then allowed to be exported only when it's mature and fully processed. And there's potential levels of gene regulation there. And we introduced a little hint of one as RNA splicing. So that gets done, and then the RNA is in the cytoplasm. That's where the ribosomes are that can synthesize proteins from protein coding mRNAs. So that's where that job has to get done. And so for any of these transcription factors, there's this loop where it starts out the stories in the gene, in the nucleus, and then it loops out through a story in the ribosomes in the cytoplasm, and then the transcription factor's out there, and it's got to get back in. So there's a physical cellular biology pathway of gene expression, and it can be regulated. And it's really nice to think about it in a cellular biology, physical, lay it out on your desk when you're studying story, where we start out in the nucleus with the DNA and the gene and the transcription factors regulating transcription, and then there's post-transcriptional regulation at the level of RNA processing. Then the RNA gets out of the nucleus. Then we have translational regulation, and then we have post-translational regulation happening out of the cytoplasm. All of that leads to potential blocks, like this one. It's a block. You know, it's an actual physical block. It's like a car mechanic talking about, you know, well, your gas is in the can next to your car, and there's no gas in your car, and you're just going to have to put the gas in to get the car to go, right? It's that kind of reasoning, you know, where it's matter of fact, cause and effect, physical reality reasoning of the, you know, the FOXO protein is not doing its job. So we learned about that idea, and after we learned about myogenin, the next transcription factor we talked about was the estrogen receptor, because that's dependent on the hormone. So we have this loop of expression, and we have that example, you know, regulating the activity of the transcription factor by physically sequestering it in the cytoplasm away from the target genes. And then we have this one of estrogen receptor where, yep, it gets made, the protein's there, it's actually binding and repressing target genes in the absence of estrogen, and then when the estrogen shows up, it does its job. And that provides this loop that's regulated. And it could be regulated by events inside of the cell. What's happening inside the cell? Like this story, it's regulating FOXO like that. Or it could be outside the cell. Oh, it's some hormone. And then it's, in lots of cases, hormones are dependent on the external environment, right? And some animals go through reproductive cycles that are based on the season of the year, based on cues like daylight. So we can have external factors, and they are involved in these loops of regulating output of the genome, like that. So that's what's going on here, talking and adding the nucleus into this story for us. Back when the authors were writing the book, it was chapter 10, and they were still building up. So you don't have any of this higher level feeling of, oh man, we have lots of examples of gene expression and we're plugging them back into these cell biology stories. Yep. So that's what's going on. And we talked about how you could tag FOXO back, toward the start of our course. You could tag FOXO with GFP, and you could do that by making a transgene where the two coding regions were together. And then we could use that to monitor the physical location of FOXO in the cell. So some good cell biology there where we're saying, oh, I see it's going. And I don't know what the specific example is here, but it's not using FOXO or GFP. It's a story, though, of the same concept that we did. And all that comes back to this thing that I was just describing that you learned about in intro bio. So this course is the first semester of intro bio, but just stepped up. And that's a central concept in intro bio, is the physical organization of the pathway of gene expression in eukaryotic cells like ours. Yep. So gene expression actually has many steps to it. And any one of the steps could potentially be the rate limiting step, now in this case, of the function of the gene. The FOXO gene is regulated at the level of transcription. We didn't learn about that. We learned about the regulation of the FOXO gene product at the level of post-translational regulation by phosphorylation and binding and keeping it out of the nucleus. So we're adding to that without losing track of our favorite story, and that is the regulation of gene transcription. Yep. Remember also way back, we were building up this list of transcription factors and how they were regulated. So we know a bunch of transcription factors that are regulated at the level of phosphorylation, and some are turned on, like alkyd and so forth, and some are turned off. And we learned especially about transcription factors, but any protein could be regulated that way. And so we learned about that some when we learned about the cell cycle. There were plenty of proteins in the cell cycle story that were not transcription factors. The CDKs and so forth, not transcription factors, that get regulated at the level of phosphorylation and other ways of post-translational regulation. And so what happened in history is molecular biologists started out in the 1940s, and they discovered that DNA is the primary heritable molecule. And in the 1950s, they figured out the structure of DNA, and then in the 1960s, they figured out the pathway from DNA to RNA to proteins. And they also started to figure out this idea that the cell has all the DNA, but it's not using all of it. So molecular biologists got very interested in the regulation of transcription and transcription factors. And then they're like, even in the 1960s, like, that is still kind of a boring robotic story, right? How do we get life and all the fascinating, features of life out of a thing where you just like make a transcription factor, activate a target gene? And the answer is, you modulate the activity of the transcription factors. And so then molecular biologists got very interested in that, because it's a really cool way to regulate transcription factors. And we learned that these receptors from chapter 16 and the signal transduction pathways, they do more than just regulate transcription factors, but they do regulate transcription factors and gene expression. Right? So the PDGF receptor in our skin cells gets activated under a scab by PDGF, and its function is to cause a genomic response in the skin cells to activate new gene expression, to speed up the cell cycle by stimulating start and rolling the skin cells fast through the cell cycle, so we could get that wound healing. And there is cellular and tissue and ecological relevance for getting that done. So there would be a natural selection that would favor that. It's a dangerous game. What's happening under a scab? It's a dangerous game. We're stimulating the cell cycle in an organism that has to be careful to avoid uncontrolled proliferation, because that's cancer, you know? So there's like natural selection at play. Is it a good idea? Would it be a selective advantage for this organism to speed up the cell cycle and heal wounds quickly? And the answer is yeah. Because, you know, when the organism has a wound, it's vulnerable, right, to having it re-wounded or not being able to fight as well or move as well because they got a big old, you know, scab like that, right? So it all ties together like that in this most, you know, like matter-of-fact way across the whole spectrum of biology, okay? So we're looking at that, and we learned about the nuclear lamins, and we're like, you know, they're pretty boring. You know, we learned they get disassembled during mitosis in lots of species. But then we get this idea that actually mutations in that could cause weird, surprising trouble, like greatly increasing the rate of aging. So we went through that and talked through that, and again, we started learning a little bit about RNA processing and RNA splicing that occurs on that pathway of gene expression. Great. And then we started learning about these, and we keep this simple. You know, it's an energy-dependent cycle that involves this import and export like that. Great. And that's that. And just more information about that, and the idea of these import and export signals. And then, you know, here's that list that the authors have of a variety of different import and export proteins. And we don't need to learn the other ones. Just the idea that, oh, wow, there's a big bunch of stuff going on in regulating you know, entrance into the nucleus and exit from. And so for us, we really like that because it adds to that notion of how transcription factors are regulated. And that's just an entry door into the cellular biology of getting things into and out of the nucleus. But it's a memorable one, you know, to just keep focus on transcription factors. And so we talked about that. We don't need to understand that one. That's a different transcription factor, NF-kappa B. That's a really important one, especially in immune system responses like activating the immune system. And we're going to hear one more time from Sam on Tuesday, next Tuesday, right? And it's going to be more clinical stuff and kind of a little bit more connecting it to real world experiences with that CAR T-cell therapy. So instead of featuring this, we featured FOXO. We really like the way FOXO can get in and out of the nucleus. And then we said, then that 1433, you know, we just said, whatever, it's got a kind of a goofy name, and it's just doing that simple job. But then on Tuesday I said, actually, it's a big family of proteins, and they do that. They do that, but they do that in almost every different kind of cellular process. There, that 1433 protein is doing that. Let's just keep you out of the nucleus. Yes?
So, actually, you just answered my question. So the 1433 is just going to hold it in the cytoplasm, anything that needs to function in the nucleus. But it won't, like, if it catches it before it goes into the nucleus, that'll hinder it, basically?
Yeah.
Okay.
Yeah, and I don't, you know, like, I don't know if there is more to the story of 1433. It's a gene family that does that.
I'm just wondering, like,
Yep.
if it holds on to it long enough, something degrades it like we saw, like with P53, or like how that...
Oh, yeah, I don't know.
Okay, we
Yep.
just don't worry about that.
Yeah, yep, and you know, maybe, well, even if it's just keeping things sequestered in the cytoplasm, that can have like a different twist to it. Like we already learned, well, it keeps CDC25 in the cytoplasm because its target is CDK1, which is mostly, almost entirely in the nucleus. So that's not a transcription factor story. So there could be these other stories that it's doing stuff like that. And I imagine there's more, you know, like as you get into it, you're like, oh, okay, I see that how that can be used in this, you know, protein family, 1433 family, to actually do more kinds of regulation.
It just doesn't seem like it could be a loop, because like if one molecule has to hold one, then it's like that you would have to have multiple. Like it's not like it's holding it, degrading it, going back and grabbing another, like we've seen.
Right,
other
yeah,
things.
yeah. No, there's just a big pool of 1433 that can be activated and bind all the ones it needs to bind. Yep, yep. And then just some slides, because I just left these in here, because if there were questions about this idea of, yep, that 1433 also does that to the CDC25 story. That's how that gets regulated like FOXO. And then we were looking at this, and we worked our way through most of this, and I just wanted to point out that there's this idea that scientists have, and we'll talk about this after we finish talking about chapter 10, and that is, well, why don't we learn about all the interactions, like Anna just asked, what are all the interactions that 14-3-3 protein family members ever have? And they just made up a cute name, they called it the interactome, like learn about all of them. There's the genome, that's all the DNA information. What about calling that the 14-3-3 interactome? And we care a lot about that because that's what the function of that protein is to interact, to bind to these other proteins and hold on to them. And and then there's just an instrument, a mass spectrometer that plays a big role in studying proteins, and we'll talk about that a little bit after we finish chapter 10 and move on to some topics that are kind of scattered in our book. And so rather than try to find the place in our book, we'll just discuss them in class, okay? And then the last little sentence in that paragraph about 14.3.3, merging 14.3 regulatory mechanisms with a focus on post-translational regulation of 14.3.3 itself, and then these interactions that 14.3.3 has in regulating the post-translational activity of other proteins. So we talked about all those things, and here's just an image, and maybe it's not the best image, but I'll show you a second one. But it's just showing here that this half circle protein is the 14.3.3, and it's binding to phosphorylated targets like FOXO. But see, look, it included here RAF. We know what RAF is, right? That's a MAP kinase, and that RAF, MEK, ERK pathway. So here, well, we think, RAF functions in the cytoplasm. So what's going on there, right? It doesn't need to get into the nucleus to do its job. And you might remember that it's actually bound as part of a scaffold. So we talked so often about, RAS and then RAF, MEK, MERK, ERK, like that. And those RAF, MEK, ERK are all bound together on a scaffold to get them to just be close to each other, right? And so here, the 14.3 is just preventing RAF from being added to that physical scaffold.
But if you prevent RAF, you prevent ELK from entering the nucleus, so it would be like a roundabout way to do its traditional job.
Right, and what all this is saying that it's just a little, it's not, obviously not in our book, and it's just a little sidelight to show this idea of 14.3.3. It binds and it blocks things from getting in the nucleus. Well, it also binds, it just blocks things, right? So here it's blocking RAF to get from getting added to the scaffold, and it just blocks its activity. And it just provides another way to remember what he said, it's part of a signal transduction pathway, and the purposes of those are to amplify, integrate, and provide a context. So this just shows how that RAS, RAF, MEK, ELK pathway has more flexibility. Because in some cases, it's RAF that's getting regulated in that pathway. What does that mean? In some cases, it's RAF that's the rate limiting step. And it's because 14.3.3 is hanging on to it. So it's showing that, then it's showing that bad, and you didn't need to learn about that, but that's one of the BCL2 family members that's pro-apoptotic. So it's showing how it also is doing something in the cytoplasm to block the pathway of regulating apoptosis, yeah? And so just, a little visual of that, not a huge deal, but just that we want to introduce this idea that at our start of our course, that 14.3 was kind of an afterthought. We were focused on FOXO. But then you go, whoa, this 14.3.3 is a fascinating family of these regulators, and they're all binding like that. Yep. And so here again, you don't have to see this, but it's from that same book, our review, and it's saying here's a role in a cell, and it's a role for the 14.3.3 protein family in insulin and glucose storage, and so diabetes, and just overall metabolism. And then something we've never mentioned in our course is if a cell, like a cell in our body, or a eukaryotic cell, it isn't getting enough oxygen, it goes into a state that's called hypoxia, and it's going to try to survive that. And these hypoxic conditions are very significant in medicine. So when a person's having a heart attack, they have a block in the blood vessels that are supplying oxygen to the heart cells, those heart cells go into hypoxia, and they might not survive. There could be recovery from hypoxia if you can get blood flow back to a system. And so that's just another example of a really important sort of cellular response. Then this is oxidative stress. It has some real strong similarities to what we learned about heat shock. And then here's DNA damage, right? And I'll just show it again. If you're not close enough, you don't have to see these. But, it shows that, horseshoe-shaped 14-3-3. And it's binding to three things, like it's binding to PI3 kinase up there, right? So it's binding to things that we know about, It's there, that's on that pathway. It's binding to FOXO. We learned about that. And there's that BAD and BAM in that case, affecting regulation of, it says caspase activation. And then over here, maybe less familiar terms, but there's that TOR that we learned a little bit about. And down here, there's going to be that PI3 kinase pathway and FOXO again with that oxidative stress. I don't think there's C-June, and we learned a little bit about that pathway maybe. And then here's the DNA damage. So we got CDC25, we got P53, and CHEK1, and there's kappa beta. And so all these pathways we learned something about, we're not going to go back and add 14-3-3 to all of them because we have a really good example. It's FOXO, And it's not that 14-3-3 always does just that. It always doesn't just exclude transcription factor from the nucleus, but it opens the door for us to appreciate, yeah, I know something about 14-3-3. And maybe some of you have learned more about it in your term paper because you ran into a receptor that's using a pathway and they're like, it's regulated by 14-3-3. Yep. So just to show you that, right, because we're getting near our end of our course, right, and the course was to frame foundational understanding of these ideas, and then you build on them like that as you go out into the real world and find more stuff, okay? So that's what I wanted to say about that big part of the chapter 10, is really focused on the nuclear pore and what that adds to our understanding of regulation of gene expression and regulation of programmed cell death, right? Yep, especially for us. And yes.
So 14-3-3 binds to a phosphate, but it seems like it doesn't have to be two. It can be like one phosphate or two, just based off of that last diagram.
Yeah, what does it bind to? And I would say it binds to phosphorylated proteins.
Okay.
And but yeah, probably a variety of different types. Like, could it be one phosphate, two phosphates? Okay. Probably. Yeah, I don't know. So then these are the other two pieces, and I already mentioned that they're described in chapter 10, and they're described in that cell biology context. We really have to look at the cell, and this chapter is about the nucleus. So we really have to look at the nucleus because we're going to see some three-dimensional organization of these. So one of them is the chromosomes. The chromosomes are inside, and remember we learned that there are these four famous phases of the cell cycle, G1, S, G2, M, and that historically people just called G1, S, G2, they called that interphase. Because when you look under a microscope, you really only see those two phases. You see interphase and you see mitosis. And the difference is, during mitosis, you can see the chromosomes, and during interphase, you cannot. And we know the difference is that they are always wrapped around nucleosomes, but the histones get phosphorylated during M phase by CDK1 and POLO and Aurora. The histones get phosphorylated, and they just fold the DNA and the chromosomes more tightly, more compacted, and they become visible. So all the time during M phase, you can see the chromosomes in eukaryotes during the mitosis, and then at the end, they get unraveled. And so this part of the chapter 10 is, okay, we can't see the chromosomes during interphase when the cell does, all of its living and, and so forth. But are they organized? Do they have some organization? So that's the next part of this chapter. And they do. And it's very, for us, the take-home message is very simple story. And this just shows the image of trying to identify the chromosomes in location in the nucleus. And there's a lot of this work done where they'll tag different proteins or different parts of the DNA with a fluorescent tag. And they'll ask, well, where are the chromosomes? We can't see them, but we could tag them and we can see where they're physically located. And so then this is just a map, okay? And you'd see it's just a hugely complex, fuzzy thing, right? And you're like, what is any value of this, okay? And what it's showing is that we'll be able to see where the chromosomes are, and we'll be able to ask questions about if there's organization inside the interphase nucleus. When we see the nucleus during interphase, it just looks like a hazy ball. But what's the organization like? And that's what the second part of the chapter is asking about is, well, where are the chromosomes, and is that relevant? Is that got some biological significance? Then the third part that we'll get to is also inside the nucleus, looking at like the organization within that, three-dimensional structure inside the cell. So it's all cell biology. And we learned, and we'll just look at this, we learned that when genes are expressed, the DNA is going to be more open and unfolded, and it's called euchromatin. So transcriptionally active genes are organized into euchromatin. And then if they're off, they tend to be packed away, and that's closed chromatin or heterochromatin. And so what the book is showing here is that there's some actual physical localization of euchromatin and heterochromatin in the nucleus. And what the message is going to be is that the heterochromatin tends to be inside the membrane of the nucleus, bound to the nuclear lamin. So inside there's a lot of heterochromatin. And then inside the nucleus, in some other structural areas, there's heterochromatin concentrated there. And then in between it is this space where there would be more gene expression. And so when genes get turned on and off, they actually physically move their kind of location within the nucleus in general. The next thing to say about this is that we want to just talk about two terms. So we'll go over here and sort of add to our terms. So we have that open chromatin, and that's called euchromatin. And that's transcriptionally active. And people say that, and what they really mean to say is that those genes can be transcribed. They're unfolded. We know they might not have the right transcription factor. So they call it transcriptionally active, but they don't really mean that. They mean it's unfolded and ready to be transcribed. Yeah, but that's always how people say that. And it's just, could be turned on and off, but it's open, ready to go. And we know sometimes that's the rate limiting step in gene expression, sometimes it's not, right? And right, for MyoD, we learned the rate limiting step, myogenin, we learned the rate limiting step was MyoD had to recruit tau binding factor. The promoter needs these accessory proteins to be lined up there to help RNA polymerase get started. And for myogenin, the missing rate limiting step was, tau binding factor can't quite make it. So myogenin helps it. Well, that means that the myogenin gene, it's already open chromatin. It's already euchromatin. It's already supposedly transcriptionally active. It's not. So, kind of an unfortunate term. It just means ready to be transcribed. And in some cases, it's ready to be transcribed. And then we have the other category, we'll just use a different color because we got them. The other one is closed chromatin. And
Excuse me.
that's called heterochromatin. And this is where we have now these two other terms we want to add here. And just to add a little bit, and that is, we could have what's called constitutive heterochromatin. Constitutive, maybe there's not another I there. Constitutive heterochromatin. And constituent heterochromatin means permanently condensed.
That's kind of like the centromeres, right?
Yep. So we'll just draw, a famous drawing of a chromosome looks like this, where it has a center, and the centromere is at the center. And then it has the ends, and those are called telomeres. And good examples of constitutive heterochromatin would be here at the centromere. There's a bunch of DNA that doesn't have any genes. It's just kind of structural to be able to hold that center point. That's what the microtubules are going to bind to and pull apart during anaphase in mitosis. And then the telomeres also tend to be constitutive heterochromatin. And there are some other regions of just permanent heterochromatin scattered around. So we could just draw those out here without labeling. So there'll be a patch here. And it's just regions of the genome that just have really no genes or almost no genes. There are very rarely genes that are found in constitutive heterochromatin because it's so packed away. It's so super condensed all the time. And that's that. There's some of that, and it's just really folded away. And then the other term we want to learn is the one that we've already been using, and that's this term called facultative heterochromatin. And facultative means it's closed, but it could be opened, it could be closed, could be open, could be closed. And so like the estrogen receptor target genes are all facultative heterochromatin. In the juvenile female vertebrate, those are packed away, and those are facultative heterochromatin. They are heterochromatin, they're packed away, but they can be opened up, and they get opened up. And then in all male vertebrates, the estrogen receptor target genes are all facultative heterochromatin. They're folded away like that. Yep. So just adding those terms, and they're weird terms, but they're actually just odd general English terms. They're not really a scientific term. And constitutive just means constant, and facultative means depends. Could be open or closed. And so we want to be aware of that because in this figure, what's out here are transcriptionally active genes, and they mean euchromatin and facultative chromatin. So that when we have the estrogen receptor target genes, they're out there. They're not constitutively packed away and placed there. Another way to think about this is that right here, that tends to be the centromeres and the telomeres. So you can think of the chromosome as kind of like odd kind of loops of structural organization. And they're just showing that. They're just showing it, the fact is inside the nucleus, we see during interphase just a grayness, very smooth, uniform grayness. Is that what it's like inside the nucleus? And the answer is nope. There's these organized regions of the nucleus, like that. Yep. Okay. So that's actually all we want to get out of that section. It's just the organization of the chromosomes. And they're just like, yep, they do have some organization. And the organization is the heterochromatin and the euchromatin tend to be in different locations within the nucleus. Yes?
Quick question about estrogen. I remember you said something about the charge that can go through the membrane. Is that probably the phospholipid biolate? Can it go through the nucleus too, or does it need one of the important or?
Yeah, so questions about estrogen, the estrogen hormone. And the estrogen hormone is a steroid. So it's not a protein. I don't have the structure up here, but it's like a 4-ring carbon molecule that if you were in organic chemistry, you would be like, yeah, okay, I'm familiar with the structure of that. And it's very hydrophobic. It just has a couple of hydroxyl groups on it that make it somewhat hydrophilic. But it's not very hydrophilic at all. And it's interesting because it goes through our blood, right? It goes through our blood, as does testosterone. Testosterone and estrogen are almost identical chemical structures. They're steroid hormones. They're a type of lipid that's just very hydrophobic. And so they're said to be lipophilic. And a way to introduce that idea is that if you study them just in a jar of water and cooking oil, and you take water and cooking oil and you shake it up, and the phases mix very, briefly, and you let it settle, and then the water phase and the oil phase, they separate, right? Just like that, right? And they're separating because the oil is very hydrophobic and the water is hydrophilic, right? So you can take different molecules and add it to the jar of water and cooking oil. You can add salt. Salt is very hydrophilic, right? It's got this positive charge. It's very water-loving. And you can shake that up in oil and water and let it settle out. And then you can say this. You can say, how does the salt, and the word is partition, how does it separate between the two phases? And the answer is 100% of the salt is in the aqueous, the water phase, and 0% is in the oil phase, the organic phase, okay? So you can do that with different molecules, and if you did that with estrogen, you'd see this really curious thing, which is you shake it up, you let it settle out, and like 95% of the estrogen is in the oil phase, and 5% is in the water phase, and it's exchanging. The one that's in the water molecules, they don't stay there, they kind of bounce back and forth because they're just barely hydrophilic. So they could kind of go dancing out in the water like that, and but not for long. They'll be like, I don't know what I was doing out on the dance floor. I need to get another drink, like that. And they go back to the hydrophobic bar area and hang out with the hydrophobic molecules back there that are not dancing, right? So that's what it's like, and that's what it's like in our body. When it's in our blood, it's never floating out, almost never floating free in the aqueous phase. It's stuck to the surface of something. It's being transported around like that. And so it's doing that where every once in a while it bounces up. And so it goes right through the phospholipid bilayer because it can be in the water environment outside the cell, and it can just diffuse into the phospholipid bilayer where there's always cholesterol and estrogen look almost identical. And then it can just go, diffuse further through, and certainly go diffusing through the membrane of the nucleus. It's small enough to probably just drive right through the nuclear pore, except for it's almost never hanging out as a small molecule, And so, and that's why we have a nuclear receptor for estrogen. It's because the hormone can get right inside the cell to do its job. And that would be generally true for obviously the ligands of the nuclear receptor family. So the other ones, their ligands must also be able to get into the cell. Yep. Yes.
Are there intracellular chaperone proteins?
Intracellular chaperone proteins for estrogen?
And other cholesterol-derived hormones?
I don't know. Yeah, I don't know. So that comes up. So out in the blood, there are some proteins that are referred to as transport proteins that people think are, especially their role is to help just kind of bring along other estrogen and other molecules that are pretty hydrophobic, and they just stick to the surface. But the estrogen, it'll just stick to the surface of almost any protein, because it'll find an area that's pretty hydrophobic and just be like, that's where I'm going to hang out, So that idea of, are there those kind of transport or chaperone type proteins inside the cell? I don't know, but kind of any protein could play that role, And, it would get in, just happen to be bound to, how'd you get in the nucleus? And you're like, I just was hanging on to FOXO and it came in, I don't know, like that. So definitely, they move around like that. Yep. And so that's that. Just the idea of that the inside the nucleus, the chromosomes have some organization, and it's organized based on gene expression. where they're this region of more expressed region. And then that's the last part. Nuclear bodies just mean, are there areas of specialization within the nucleus? And scientists can see them, and they can stain them. Let's just go to that kind of image. So here's just an example where this is looking at an in interphase. The nucleus just looks like a fuzzy gray ball, and that includes during S phase. During S phase, the nucleus doesn't look any different, but we know there's lots of DNA replication going on. And so if you look within that smooth gray nucleus during S phase, and you say, now, is DNA replication taking place everywhere? And the answer is no. It's taking place in these little areas that just seem like areas of specialization, like a little DNA synthesis factory, like that. And that's what they call those. They call those replication factories. And you know, so remember how we said, like, well, we want to talk about the nucleus because there's the pores and they work as gates, and we're going to have like a physical localization of this stuff, and because of that, that brings in a layer of potential regulation. The FOXO needs to get into there, right? So that's why people are studying this, is because if there are physical locations that where this activity is there a regulation about getting to that? We're inside the nucleus, so it's not the nuclear pore, but there's inside the nucleus these concentrated patches of activity, and might they be regulated? Might they be regulating that by, for example, MCM? And is MCM making it to the replication factory inside the nucleus? So questions like that is why people look. They almost always are like, structure is fine, but how does that relate to potential regulation? Yes?
Are what we see in the polarity of these nuclei the like metaphase plate? Does it
These
exist?
nuclei?
Does it exist like outside of the specific moment in metaphase?
I don't know, I'm just being Vanna White.
You're doing a great job being Vanna
Yes.
White.
Yeah,
I
you
don't
want to
think
buy
you're
a
at
vowel?
phase,
Who
you're
wants
at
to buy a
S phase, aren't
vowel?
you?
Huh?
Aren't you an S phase, not M phase anyways, though?
Here?
Well, exactly. And my question is like, are what we looking at is already the metaphase plate even outside of M plate?
I don't think so.
But I do have a
I
question
don't think
that
so.
kind of is about that.
I bet we are.
I
'Cause
bet.
Look at that. Look,
Yeah.
look at the clear polarity.
Get you, would you have cash?
No, I have Venmo.
Yeah, okay. Start Googling.
All right.
Yeah.
So can I ask another
It's
question?
$20, and you have Venmo, $20, I have Venmo, and I don't have time to Google, so you figure it out. Yeah?
So my
I
question.
probably won already.
So my question
Right?
is about the.
I'm gonna buy everybody pizza for lunch!
It's about the constrictive heterochromatin. I know you said that it doesn't have much genes, but if you're going to go through S phase, you need to still replicate that, don't you?
Sure, absolutely,
So
sure.
it's really not closed. You still need to open it.
Well,
and
but,
replicate.
so that's a good thing to bring up, is, you know, when we talk about DNA, there's really two things we want to talk about. Transcription, gene expression, and DNA replication. And it's super important to keep those separate. So when we say something has got constitutive heterochromatin, yeah, definitely it has to get replicated.
But it's just never transcribed. Like you're never
Yeah.
going to like.
Yeah.
Okay.
During S phase, the cell is really focused on DNA replication.
Mm-hmm.
So this has no relevance for expression.
Right.
And that heterochromatin story is all about, you know, expression.
Okay.
Yeah, so even though the centromere and the telomeres are pretty packed away, when it's time to replicate, the DNA polymerase comes in with a whole different set of helpers to get that going and come rolling through there and again make sure that gets
I always
copied.
think like the detangler, like the topoisomerase or whatever is like
Yep.
the detangler that opens up.
Yeah, there's a lot of helpers that are involved in that, and the book talks about the DNA replication some in chapter 5. We're not going there.
Okay.
Yep. I have a couple of figures from chapter 5 to show you, but not because we're going to read that chapter. Yep. Yeah. Did I win, Sam? You're looking like you have a way of holding your mouth that makes it look like you're losing the bet.
The short answer is Google doesn't know. This is too advanced.
Man,
So
you know what that's called? It's called the Google smack. Have you guys ever heard of that? If you can Google something and Google goes, no hits, I found nothing. That's a Google smack. It used to be a thing when Google was first getting started. It used to be a game people would play to try to Google something that Google's just like, got nothing. Yeah, you know, yeah. So I won that Google smack. Boom, boom, right? Yeah. Which reminds me, shh, I'm getting paid for this.
Ignorance is bliss.
It reminds me in 4th grade in Sunday school, our teacher was a big farmer, Mr. Gilman. No, it wasn't Mr. Gilman. It was some other farmer, but he was our Sunday school teacher. And at this time of the year in Sunday school in Minnesota, he said, you guys have been doing great. Spring's coming. I have to get out and start plowing. So next Sunday is my last Sunday teaching Sunday school. And so I'm going to bring each of you kids a quart of pop, because that's what it is in Minnesota. I'm going to bring you each a quart of pop. And we're going to have a contest to see who can drink a quart of pop fastest. And he asked us that Sunday, what flavor do you want? And Louis Gilman, so it wasn't Mr. Gilman, but it was the kid, Louis Gilman, and Louis Gilman says black cherry. And I'm like, that's a mistake. You do not want to chug a quart of black cherry soda. And 4th grade, I said that to myself, right? And we get there the next week, and Louis goes straight ahead with the farmer, chugging this black cherry, and throws it up. Yeah, yes. So that's just what happened in our class, right? That student, the Louis Gilman of your college experience, goes head to head with the teacher, loses a bunch of money, gets a Google smack down, and,
I agree with
and
him, though.
throws
Lack of
up, and
evidence
as a
doesn't
result,
prove.
throws up. And I'm like proud, because I'm like, I didn't pick black cherry. I was 3rd place. The farmer, then Louis, then me. And anyway, I'm not bitter. But yes, all right, let's move on. And so, stuff. So then the book shows this kind of drawing, right? And obviously it's a little bit of fanciful drawing to bring this concept. But then it says, here's another one. And that is, there are these transcription factories. So we saw, and we got to keep these separate, a DNA replication factory during S phase. Well, there are also these transcription factories. So that euchromatin is out there, right? Heterochromatin elsewhere. The euchromatin is further organized into these little dots of activity, right? And we want to know, well, is there regulation happening here, right? And it's an area of study, but it's only cell biology that brings it up as a potential issue, right? You got to be able to look at the whole cell and see, here's an area where you might not be allowed in or not. And I don't know any examples of that where somebody says, yep, there's regulation at the level of can you get in or out of the transcription factory. Yep, but it's just a reality that, and you kind of imagine that probably, but I just don't know. It would be an area of current research. And it's the kind of thing that, how are you going to study that, right? You got to come up with a strategy to be able to study whether or not a protein is getting access, right? So could you use that tagged FOXO to see, well, yeah, I got in the nucleus, and then was it able to get all the way into the transcription factory region, like that, just like that. And again, right, remember there's the membrane here, and that's the gate. And this, you drew a circle around it, which was a mistake, because there's no membrane, no gate here. It's just a set organized area. But maybe there's some features that make it more or less able to get into that space, right? So that's what the book is showing there. And so here, and it calls those factories, it calls those a more general term bodies. So nuclear bodies, like little nuclear places. And it wants to describe, list them, right? So it lists some here, and we don't have to learn this list. The first one we'll mention a little bit because it came up on Tuesday. So the first one is called the nucleolus, and here's, is this the slide for this? So here's this nucleolus region, and inside the nucleus is something that you can sometimes see in a eukaryotic cell. So it generally looks like a gray kind of hazy thing, but the nucleolus is such an organized nuclear body that sometimes you can see it. And species vary in the number of nucleoli per nucleus. So there might be kind of like distinctly 2 nucleoli in the cells of this species. So scientists have known that a long time because they've used that as a way to kind of distinguish between subcells in different species. Like quail, quail have a different number of nucleoli than chickens. And so they've been able to do old-time experiments where they would mix those cells together and they'd be able to keep track. Oh, that's a quail cell that has 2 nucleoli. Oh, that's a chicken cell that has 3. So that's just a little detail to help you remember that you can actually see the nucleoli often. And then, so it's an exception, and it's an example of a nuclear body. It's a specialized organized region within the nucleus. And this is what's happening there. And we talked about this a little bit on Tuesday. And it's like this. So the mature ribosome that is going to synthesize proteins that's in the cytoplasm is really made up of about 60 to 70 different proteins and then a couple of different strands of ribosomal RNA. And so we're going to have to put that together to build the ribosome. And that's what the function of the nucleolus is that's the nuclear body that is a ribosome assembly factory. And why there? Because it has put the genes for the ribosomal RNA there. So they're in euchromatin, so open transcriptionally active chromatin, and they're transcribing genes that are not coding genes, they're non-coding genes. They don't code for a protein, they just code for a big long RNA, ribosomal RNA. So they're non-coding genes, they're transcribed there, and then the RNA processing is done there, so they get processed and ready to be mature RNAs, and then they get assembled into the subunits, the 2 subunits of the ribosome there. So again, inside the nucleus, we have some nuclear bodies. The ones we learned about are DNA replication factories that would be operating during S phase. We learned about transcription factories that would be operating in G1. And here we're learning about the nucleolus that's operating sort of in G1 also, where we're going to make some more ribosomes. So we got to make them. We need more ribosomal RNA. So we're going to transcribe the ribosomal RNA genes, and we're going to have those there. And before they leave the nucleus, we'll just have them processed there, have them ready there, and we'll put together the big parts of the ribosome there, and then we'll haul that all out through the nuclear pore. So we'll export that as a big chunky thing, like a school bus. Yeah? And so that's why the nucleolus has a visible organization, is because it's such a factory of activity. And so we'll just talk about the pathways for the ribosomal RNA. So again, the DNA is for those genes is in the nucleus, and it's transcribed into ribosomal RNA in the nucleus, and they're processed there in the nucleus, and then they get packaged into parts of the ribosome that then leave the nucleus. And that's where the ribosome's work is, outside the nucleus in the cytoplasm. So just one time we get the ribosomal RNA out of there, and it leaves because it's bound to the other proteins. So what's really being exported is the other proteins, and they're just in its big assembled thing. Yep?
So those ribosomal proteins, though, those are transcribed and translated by something else.
We'll talk about the ribosomal proteins in a second.
Sorry.
But just to start with the ribosomal RNA, that's its story. It's getting out of the nucleus that way. And I'm not sure, but let's just go back to this slide. And we don't have to learn anything new from it, but I was just looking to see if they had on here, so we are just learning about importin and exportin, but it would be a different set of proteins that are helping the ribosomal units coming out of the nucleus. Okay? And it just doesn't have the name of those, and I have no idea what the name is. Okay, so that's that, okay? So, not that one. That one. Okay, so then, we also we talked about this briefly on Tuesday, but let's just mention this again. And have you Venmo'd me $20 yet?
No, you didn't ever give me your Venmo. Also, I'm not convinced you won.
Sorry. I'm stopping the recording. What is your other comment?
I'm not convinced you won.
Who's convinced I won? Who wants to pizza? All right, so then we have to talk about the ribosomal proteins, because they just have a little confusing pathway. Because the ribosomal proteins, they must have ribosomal protein genes in the nucleus. It must be in a transcriptionally active transcription factory, and those genes are getting transcribed. They are coding genes, so they're going to be coding for a primary transcript that's going to get processed in RNA processing in the nucleus, and a mature transcript will leave the nucleus, and it will be a mRNA, a messenger RNA, because it's carrying the message to the ribosome, and an existing ribosome will synthesize this new ribosomal protein. Then, for it to get to be part of a new ribosome, it has to have important help it go back into the nucleus, where it goes to the nucleolus and it gets assembled as a ribosomal subunit inside the nucleolus with other ribosomal proteins that are newly synthesized and the ribosomal RNA, and then it exits the nucleus where the ribosomes function. So it just kind of underscores, oh wow, there's a lot of stuff about traffic.
So it has the NLS?
Yeah.
Okay.
Yeah, there's a lot of this traffic to these different locations, and that's just pure cell biology, right? It's like, guess what? We got to talk about the cell, You molecular biologists sit down because you know the details of, voxel gets phosphorylated. Okay, well, we're going to tell you what that, why that matters, right? Because it can't get into the nucleus. That's a cell biology concept. So this is just a little more of that nuclear body, the nucleolus. So the nuclear bodies, again, are the DNA replication factories active during S phase, the transcription factor active during gene expression, especially G1, and then this nucleolus active just kind of most of the time. And your body needs more ribosomes when it's going to grow and stuff like that, so you have to assemble more ribosomes like that. And it's a big factory. Yeah, who wants to chug a quarter pop? Or bet me another 20 that I can guess better. Man, it's ready. I'll bet. How many ribosomes do you think a typical one of your cells has? Cam, how many?
10,000.
All right, Cam is in for losing 20 at 10,000. $20. Anybody else want to play?
Is it too high or too low?
Wow, the price is right, Anna. Yeah, you want to go like 10,001? I'd like to bet 10,001, wink, Martindale. No, it's like 100,000 and up. It's just a huge number of ribosomes. So, and they only do protein synthesis. It's a big part of our cells, okay? So then here's the only other one we're going to talk about, and it's on our list of nuclear bodies, and it's a place of facultative heterochromatin. So we're, again, remember we got the constitutive heterochromatin. Oh yeah, that's out at the edges of the nucleus and then in the center of the nucleus a little bit. And then we have this euchromatin that could be made up of euchromatin that's transcriptionally active or facultative heterochromatin. And so here's some facultative heterochromatin, so it's repressed. It's packed away, and it's packed away in a special location, and it's packed away by some proteins that are called polycomb, which is a dumb name from fruit fly geneticists, but let's just write that down. And that is, when we talked about, this open chromatin, and then we got the closed chromatin, But we've always been talking about really facultative heterochromatin. So stuff that's packed away so it can't get expressed, but it could be, could open that up. And so a subset of this, facultative heterochromin, is packed away by a family of proteins called polycomb. And so they are repressors. They're going to pack it away and just fold it up and put it away. You can't use it. And this is kind of the most famous example of a set of proteins that can put away genes, repress them, and just keep them shut down. So remember we said, oh, when a cell differentiates, it's going to methylate a lot of the DNA and keep those genes off for good. Another thing that happens is other mechanisms, like polycomb, will repress gene expression over just a big region and say, we're just going to shut down gene expression in that region. Okay? And the polycomb proteins are actually transcription factors but they don't have a DNA binding domain. They don't bind directly to the enhancer. They would therefore, we would want to call them co-regulators because they bind to other proteins, right? We would like to call them a co-repressor. And some people like to do that, and some people don't. Because polycomb, we've learned so far about co-regulators that will bind to other transcription factors that are bound to the enhancer, right? That's how they work. So beta-catenin, it doesn't have a DNA binding domain. It doesn't bind directly to the DNA. It binds to TCF, right? So polycomb doesn't bind to those enhancer-bound transcription factors. Polycomb binds to the histones and the kind of the proteins that package the DNA. So it's just another kind of type of transcription factor where it's folding up the chromatin, but not by binding to an enhancer-bound transcription factor. And let's see, we're just going to do a quick concept chat, and then we won't have time for anything else except for Venmoing people who've lost money to me during class meetings. All right, so last couple of weeks in class, we don't know how much more, we don't even know how many more classes we have, but it'd be a good time for you to say hello to another classmate and check and see if they're graduating on May 9th. Oh, we're going to do this, and I have to pen best your exams too, so get ready for that. we're just gonna do this for another minute.
I mean, they're just like Arduino cells that don't involve some right? Like, end up with a genomic response.
Okay, so now, we just have a couple minutes left, so, and because I have to also hand back for your exam. And so let me just mention again about your exam. You'll have a circled score on it. That's the score you got out of 100. Remember, I don't write down many comments on exams. Instead, I make an answer key video. And so, there's lots of things I wanted to say on your exam, but I just decided to try to put it all together. So the first thing to do is, double check that I gave you the right score, counted the, numbers wrong. And then if I didn't, just let me know. We'll double check that. And then hopefully listen to the whole answer key video as you're looking at your exam and pause and focus on the ones that you got wrong and you're like, I'm confused about that, but listen to all the answers to see if you can learn some more from that. Great. And when I send out the link today for the answer key video, also send out a Word document attached to that Brightspace announcement that shows the grade distribution. And I don't curve, I'm not heading for a 90, 80, 60 cutoff. Instead, I just do that anonymous distribution and I say these students look like they got an A, B, C, D, and I do that based on the class, but I've also taught this class and other classes a lot more. And so you can see where you fit for your grade. And so again, it's just your circled score that shows up on the grade distribution. And let me know about that. Last thing about that is the final exam is worth even a little bit more, right? Because we still have students who are like, darn it, I got to do better on these exams. And you can still improve your grade in this course by doing well on your term paper, so putting energy into that, and then also the final exam. So, you and I can talk about that and where you fit in the course. So for our concept chat, we're just going to say this for the one time this semester. We'll say who is one person that has something they want to say about something that was important in class today. Yeah? Okay, Sam.
No, I'm saying not me.
Not you, because you lost a bet, so you probably don't want to go. See, and what if I done it that way every time? We just waste a lot of time where I'd be like, who's something they want to talk about, right? So instead I was calling on people and doing it in a pairwise way so you wouldn't feel like, oh, I was putting the pressure on you rather, sorry. Yeah, see, right? So Rustam is an exam, and then when you get these, you can head out. So hang on to these.
So I'll read through it. Sorry, I've been dropping you all off.
No, it's okay. Like, you don't have to indefinitely. I just need to make sure it's
Yeah, I mean, not that I know any better, but I'll tell you that mine.
Is not
Yes.
here today.
the taster sector. You have actually a couple
Oh, perfect. Thank you.
And you have the right reason too.
Well, and I originally went wrong. Thank God I checked and I saw that this was napkin because originally was backwards.
And we'll deal with this and we'll move forward. And I'll put this 20 back in my pocket for now.
This was such a shot in the dark. I didn't get it.
Yeah, I don't know if that'd be awkward.
Oh, Rustam, we're going to talk. Should you want to just talk in the hallway or back in my office? Okay, I'll see you out there in a moment. Yeah, cool. All right.